cross-section of the villi, what would that look like versus a cross-section of the intestinal glands?
Circles.
So here we would have a space, we would have a circle that's solid in the center because of the lamina propria and the simple columnar cells forming a ring around them. So we would have simple columnar epithelia, goblet cells. We would have, you might be able to see the lumina of the lacteal, and you would have loose irregular connective tissue filling this up. Okay. That's a solid structure with a solid core. Now, if we were looking at a gland, so this would be the villus. If we were looking at a gland, that's going to be hollow in the center. The columnar nuclei are going to be facing away from the lumen. And we would have loose irregular connective tissue solid around this. So the two are reversed. Okay, the glands are hollow in the middle, the villi are solid in the middle. So this would be an intestinal gland. Which makes sense when you think about what your type of structure that you're looking at. Okay, then we're ready for the muscularis externa. We want pictures of these cross sections before I erase it. And we're back at our pattern. So smooth muscle. Remember, I want you to include the words inner and outer. We're not talking about belly buttons here, okay? And innermost when we're talking about the stomach. So an inner circular layer. And an outer longitudinal layer. On this model, because it's cut at right angles, it's showing the model, the sections at right angles. So here we're seeing smooth muscle cut in cross section and smooth muscle cut the length of the muscle cell. That's the reverse here. Here the smooth muscle is cut the length of the muscle and cut in cross section here. So you should be able to figure out whether this view is a longitudinal section of the small intestine or a cross section of the small intestine by looking at the orientation of the muscle cells, how they've been cut. So let you figure that out. And here you can see, so this is an inner circular layer, is our small intestinal structure. Would this be a cross section, or would this be a longitudinal section of the length of the smooth muscle of the small intestine? It's a cross section. Okay. So, smooth muscle cells that are the inner circular are going to be cut the length of that muscle cell as they form a circle around the lumen. The longitudinal cells would be coming out at you. So when we cut them in cross section, we cut the cells in cross section as well. If that seems fuzzy, work with the model during lab, okay? Because that tells you the orientation. So here we have the submucosa. Now, notice those glands. So where do you think this has been cut? All you can see are those glands. Those are not intestinal glands. They would not be that close to the inner circular layer. So they are glands of the submucosa. No, we're small intestine, my dear. So where do we have submucosal glands? In the small intestine. And the duodenum, exactly. So even though all you can see is that little corner up there, because those are submucosal mucus secreting glands, we only have them in the duodenum. Okay? So then we have our muscularis externa with smooth muscle in the inner circular layer, longitudinal muscle in the outer layer, right? And we have remember we talked about the enteric nervous system of the small intestine when we did the autonomic nervous system last semester. We'll talk more about this on Thursday. We have these autonomic nerves in the muscle layers. The myenteric plexus is another name for it, and the helping to control the peristalsis of the smooth muscle. And then out here is our serosa, or our visceral peritoneum. So we have a loose, irregular connective tissue, and the mesothelium is just another word for the epithelium of the mesentery. Some of you may have seen ads, or there are always these ads from lawyers, individuals that have a type of cancer known as mesothelioma. So that's referring to, it doesn't necessarily can be in the lung tissue as well, and the mesentery, the pleura of the lung. It doesn't have to be the abdominal system. So that's our last structure then, is the serosa, which is the visceral peritoneum, loose irregular connective tissue attaching it to the muscularis mucosa, and then an outer simple squamous epithelium. So this is a cool slide. You can see everything except the mucosa. So back to the mucosa and submucosa here. Because of the importance of the small intestine, we talk about 3 orders of folding that help to increase surface area. Because this is our primary organ for complete, final, intermediate, and final chemical digestion. So we need the surface area for the enzymes that the small intestine synthesizes, and we need surface area for the absorption of the products of that chemical digestion. And so we have a series of foldings. I'm going to erase the board here in just a moment if you want pictures of those additional notes. First of all, we have folds of plasma membrane, which we've described as either cilia or microvilli. And in this case, they have a core of actin. They're not motile. We're not trying to sweep the mucus along. And so they have a core of actin and create an increased surface area through the microvilli structures. Folds of plasma membrane. And then we place that epithelium, that simple columnar epithelium, we take that and create a fold of that epithelium with the underlying connective tissue. This is like taking a tree with a bunch of leaves on it, and how can we get more trees? We can create a hill, all right? We can put more trees on a hill than we can, say, in a square mile of flat land. So if we have a square mile, corner to corner, like if you fly over it, you have mile mile, but then we have a hill in the middle of that. We're going to have more ground. It's going to take you to walk longer to walk over that hill from corner to corner than if it was flat. And so by taking this fold of mucosa, the epithelium and the underlying loose irregular connective tissue, we've increased our surface area. And now something that is unique to the small intestine. We take the submucosa and shorten it up and create folds as well. So remember when we talked about peristalsis? We were talking about our muscularis externa bunching up the digestive tract. Well, we have almost a permanent bunching up of the submucosa lining the small intestine. So if I were to take my sleeve here and I pushed it up onto my elbow, over my elbow, notice how the fabric has folds that go around my arm in a circular fashion. These are known as circular folds. They are visible in the small intestine to the naked eye. So here we have the duodenum and jejunum, pyloric sphincter, and notice these ridges that run in a circular fashion lining the lumen of the small intestine. Those are the plicae circularis. So I'm going to take one of these folds. Notice this right here, the structure. That's the circular fold that's been cut in cross-section, transverse section. So if I were to take that, so again, this would come out and around to create a ring. This is submucosa here. So rather than drawing that in black, let me draw that in red, because that would represent our muscularis mucosa. So this is a fold of submucosa, and then on its surface we have our villi and intestinal glands. So now we can have even more villi by taking our fold of submucosa and there would be more than one gland. I'm just trying to hurry things along here. So this would be a villus. And we would have little microvilli all over those. So you can see that we've increased the number of microvilli we can have by having a villus, and we've increased the number of villi we can have by having a fold of submucosa. So this fold of submucosa is identified as a plica, plicae for plural, circularis, means circular fold. If you had some photos that you printed off, and I know that seems redundant because we all send them by email and text and message and all that kind of stuff. But my mother still hasn't quite figured this out. She'll take Facebook pictures that my niece has posted from Australia, have my dad print them off on a photo printer, and then mail them to me. It's like, Mom, I get Facebook, I have the same pictures. And she'll print off letters that she gets by email that I get and mail them to me as well. So if you wanted to put a photo in an envelope and write "do not fold" on it, you could put "do not plicate." Of course, nobody would know what you were talking about, so it may not save you anything, but that's what the word means, is fold. All right? So this would be the submucosa in the center and the villi extending out from that surface. So here's another view. Here's that circular fold, and this would be the section through it that we see as a plica circularis. So now on the screen, these are individual villi. In fact, right here, you can see a wide space channel within the villus. That's the lacteal. Here would be the surface epithelium, columnar epithelium with goblet cells. right? This is an extremely wide lacteal in there. So this is similar to what I just drew on the board. Muscularis mucosa would be right through here. This would be submucosa. So this picture image is a picture of a section through a plica circularis. Okay, any questions on that? Be sure when you're looking at the microscope today that you pay attention to comparisons. Don't just look at one region of the small intestine because it moves in the, it's cut in different angles because it's folded in different ways. And so in one section, you may see something like this where you're looking in the intestinal glands. In another part of the tissue, you may see cross sections through the villi themselves. So move along the side until you see an area that is recognizable to you.
You said the submucosal glands,
secrete mucus
into
into the duodenum, so it sticks on top of the villi there and protects those cells from the incoming acidic material from the stomach. All right, that brings us to the large intestine or colon. So I want you to be comfortable moving back and forth between those terms. If we talk about the ascending colon, we don't talk about the ascending large intestine typically, but the terms large intestine are utilized. And so that brings us to our next sphincter. And so between the ileum and the colon, or cecum, we have the ileocecal valves. It's a slit. It's still a sphincter, so if you call it a sphincter instead of a valve, I'm okay with that. It's located at this area here where the ileum attaches, and we have, it sounds gross, but we have a part of the colon that's been rinsed out, not like our female, it still has plasticized fecal matter in it. So here is, you can see the plica circularis here of the ileum, and the little window's been cut open. And this is the blind pouch of the cecum. A window's been cut into that, not so it can see, but so you can look in. And the pen here is on the lip of the ileocecal valve, right? Typically, we have the appendix, and this one's rather long. It's usually more often than not, folded back onto the posterior aspect of the colon instead of hanging down from it. So this is what it is. It has its own little mesentery, and it's sticking up here. So I'm going to pass that around so you can take a look at it. While you're looking at it, I haven't talked about it yet, but I'm going to point it out now. You'll notice that the colon here has little pouches that you see in this pattern. Those are called haustra. And it's especially clear on the front. There's about a 1/4 inch band that you see posteriorly. The posterior band leads directly to the appendix. There are 3 of these bands. They are identified as tinea coli. And they are outer longitudinal smooth muscle. That is reduced in the colon to these 3 bands. So take a look at those as you guys pass this from one to another, and then this will be available to look at during lab as well.
the appendix, is that?
The vermiform appendix,
Is that different from what is removed when you have an appendicitis?
no, same thing, appendix and vermiform appendix. Vermiform is worm-like, so it's that same structure there, and it has its own little mesentery here. Now, before I diagram this on the board, we're back to our peritoneal, intraperitoneal, and retroperitoneal components. The ascending colon is retroperitoneal. The transverse colon has its own mesentery, so it's intraperitoneal. Then we're back to retroperitoneal again with the descending colon. Our sigmoid colon has its own mesentery, though our models never show it. So it's intraperitoneal. And then our rectum is draped on, the superior rectum is draped with peritoneum, but the rectum is in the pelvis. Okay, so the rest of the rectum doesn't, we don't talk about as being intra or retro, it's inferior to the peritoneum. So this would be the ileum attached to the colon, and this would be our ileocecal valve. So let me draw the other one. So oftentimes, if someone is having exploratory abdominal surgery for some particular reason, they'll go ahead and take the appendix out, whether it's inflamed or not, while they're in there. A few years ago, I read an article from a British journal in which the supposition was made that the purpose of the vermiform appendix, which looks like the Peyer's patches if you look at a cross-section, it's filled with lymphatic follicles, its purpose is to store the particular E. coli that you have in your gut, in your large intestine. So if you had severe cases of diarrhea or dysentery, an unfortunate trip to Mexico or some other country where you drank the water, that this would be able to repopulate your bacteria to your colon. Now, newborns populate their colon with bacteria from the sterile appendix. So I'm not sure that that's really a necessary. I suppose it could happen, but I don't know that it's a necessary event. I had a friend who went to medical school in a Caribbean medical university, and he didn't want to have to drink bottled water the entire time that he was there. So he took the local water and froze it into ice cubes, little ice cubes, and then gradually introduced this bacteria to them by adding an ice cube or so over time to try to get used to it. I don't know that it ever worked, or if he just had dysentery for months at a time, but that was his mechanism, because it's, nobody else, it's just when we go visit countries that we don't, everybody else is drinking the water. So obviously the colon has E. coli bacteria. As a function of the colon, as you'll see when we look at the histology, we're no longer doing chemical digestion. All right? So the purpose of the colon is primarily an absorptive function. So what do you think we're absorbing?
Nutrients and bile.
Water.
Most of our digestive nutrients are absorbed in the ileum, so it's primarily water. And then we have a symbiotic relationship with the bacteria. They break down the plant fibers that we can't see, cellulose, since we don't have that enzyme, and they produce vitamin K that our liver utilizes in the synthesis of clotting factors. Always write my words in here, right where I want to draw my organs. So we have the ascending colon. Transverse colon. Descending colon, and then this kind of S-shaped region is the sigmoid colon. We have a couple of bends, actually the left side is slightly higher. We have ligaments. Kind of an attachment here. And these bends are known as flexures. So this is the right collet flexure because the liver is over here. It's also known as the hepatic flexure. And we have the left collet flexure, and because the spleen is on the left side, it's also known as the splenic. Before I add additional structures, what would be the blood supply to this area? What would this vessel be?
The superior colon.
Superior, not colon. Superior mesenteric. It gives off branches as the right colic and middle colic. And of course, we've cut the branches to the small intestine here. And then over to this side, we would have the inferior mesenteric. And giving off the superior rectal and left colic, supplying the descending colon. What is the nerve supply for parasympathetic following the superior mesenteric pattern? Same as the one into the stomach and the small intestine. Parasympathetic component? Vagus. And it stops at this flexure right here where we transition from superior to inferior mesenteric blood supply, and we pick up pelvic splanchnics, okay, for our parasympathetic component. So the pouches are known as haustra or haustrum for plural. Again, we see just that one of three bands of the outer longitudinal, which is the Taenia coli. There are these little bags of adipose tissue that hang down. They're not on your lab list. I'm not going to ask you to know them, but they're the epiploic appendages. So we have a mesocolon for the transverse, suspending it from the posterior abdominal wall. Remember, our greater omentum is going to be attached to the transverse colon as well. So to see the transverse colon, we have to lift up the greater omentum. So this is our transverse mesocolon. And then we have a sigmoid mesocolon. That's not on your lab list because it's just really difficult to find the structure that illustrates it very well. There's the blood supply, drainage. This is just an illustration of the changing epithelia from our villi to our colon, from duodenum to jejunum and most distal aspect. So here's the histology of the mucosa. Remember the plan, the schematic that I gave you for the stomach with its gastric pits, and the small intestine with the villi and intestinal glands. So now we lose the villi, we lose the gastric pits, and then the large intestine and the rectum, we just have intestinal glands. No other specializations. So take a picture, if you want to, of the large growth structure of the colon, and we'll look at the GPAP. Bye.